So our next speaker is Bob Goldberg from the Department of Molecular Cell and Developmental Biology at UCLA. Bob is a real pioneer in plant molecular genetics and genomics. He was one of the first people to recognize in the 90s the power of genomics for agriculture, and was one of the co-founders, was the co-founder of Ceres, which was an important genomic technology company for plant science that has morphed into one of the major biofuel seed companies. Bob's scientific passion is to understand how plants make seeds, and he'll talk to us about that today. Louis Wolpert famously said about animals, it's not birth or marriage or death, but gastrulation, which is truly the most important time in your life, which adds to the sense that many of us have as you get older that it's all downhill from. I didn't realize it was downhill from gastrulation. I thought it was kind of college. But for many plants, it's the making of the seed, and that's what Bob is going to tell us about. As you'll see, Bob is an outstanding educator. He has an enormous number of honors that have been given to him for his teaching. He's constantly turning up on lists like the hottest courses in America. He's a Howard Hughes University professor to develop new teaching methods. And as you'll see, he's embedded in the Hollywood culture in UCLA, and so he's going to have a lot of great audio visuals for us. This morning, Bob's going to tell us about how plants make seeds using genomics to dissect seed development.
Well, thank you very much for inviting me to be here. I hear all about JGI and all the amazing things that are done, and it's really nice to be able to talk at a JGI meeting. I'm going to change tones just a little bit this morning and talk about a passion of mine, which is seeds. But before I go into some specifics, and it'll really be a cartoon of a lot of things that are going on inside my laboratory using genomics to understand seed development, I think it's really important to point out that we have a really big problem over the next 30 to 40 to 50 to 60 years in producing food. And in fact, as you can see from this slide, population is increasing tremendously. And because of that, we're going to have to probably produce more food in the next 40 to 50 years than in the entire history of humankind. And more importantly, we're going to have to do that with decreasing amount of land for agriculture. Many of you are going to be very, very surprised to know that in Los Angeles County, California, United States, between 1930 and 1950, there were 30,000 farms. And in fact, Los Angeles County was the biggest agriculturally producing county in the United States in that particular period of time. And clearly now there's absolutely no farms whatsoever. So we're going to have to produce a lot more food on a lot less land. And that means those of us who are doing things related to agriculture have some major challenges. And clearly one of the challenges is to be able to increase yield, which is the key, being able to make more plants on less land, and to be able to use genomics and genetics and state-of-the-art science in order to maybe increase the number of seeds, get bigger seeds, more seeds, but clearly engineer the plant, as been done over the last 10,000 years, because all the crops that you buy in the grocery store were engineered by our ancestors many thousands of years ago. But to be able to use genomics in order to find genes that can increase yield and improve yield traits, but also clearly be able to mitigate abiotic and biotic stresses. These are really major problems in agriculture. And by then using breeding and genetic engineering, which is really an important part of the toolkit, to be able to increase yield, to make more plants, more food on limited resources and a smaller amount of land. Now clearly seeds are very important in all of this, because if you look and see of the major crops, the majority of them are seed-bearing crops. And in fact, directly or indirectly, over 75% of the calories that we consume and animals consume come from seeds. And so seeds are extremely important, not only for growing plants, because all plants derive from seeds for the most part, but from a nutritional point of view and a caloric point of view, seeds are critical for human consumption. And so clearly, the focus of this talk is going to be soybeans. And one can ask the question of why one would even bother with soybeans. Well, In the United States, it's the second major crop behind corn. And it's very important as a major food source, tofu and all kinds of things, but mainly as a source of food for animal feed, chickens and cows and other kinds of things, but we use it for human consumption as well. And a very important source of oils, clearly, some of which are used, about 20% of the oils from soybean are used for biodiesel. It's also really an excellent plant to study seed development and plant development. The soybean genome has been sequenced thanks to all the folks at the JGI and others throughout the United States. There's a lot of history and gene expression, as I'll show you, and clearly it's also a good source of getting funding to be able to study some really interesting and important questions. So I'm not going to go into a discourse on plant development or seed development, but it's important just to understand a couple of things. First of all, there's really three major periods of any developing seed, and that is in the beginning where the seed differentiates all the tissues and organs of the seed, so it's a period of differentiation and morphogenesis. And then the seed completely switches in terms of its developmental program and goes into a program where it's accumulating all of the storage reserves that are going to be used during germination until the plant gets up and starts photosynthesizing. And this is the source of all the food that we eat, whether it's in corn or soybean. This is the period at which the genes which are involved in making the food and the food reserves are produced. And so from a human and animal consumption, food consumption, this is really the most important period of seed development. And then finally, of course, the seed goes into dormancy. Some seeds can germinate after 2,000 years, so they can have a very long period of dormancy. And in fact, then after this dormant period, then you get the plant being made, you get the seedling. And so it's differentiation, then a maturation where you're just producing a lot of storage reserves. There's no differentiation going on, a lot of cell division, a lot of cell elongation, and then dormancy and germination. So I'm just going to give you a cartoon today, very, very, very short synopsis of some of the things that are going on in the lab to try and understand in a holistic way the processes involved in seed development, with the ultimate goal of trying to understand the genes that are critical for making a seed. And so I'm going to talk about some data on characterizing gene activity in every cell, every tissue, every organ, every region of a seed from fertilization to dormancy using extensive transcriptome analysis. And then I'm going to talk about some very new data which attempts to do the same thing, only addressing epigenetic questions, and that is characterizing the methylome throughout all of seed development from fertilization to dormancy and in all the different parts of the seed. And so how did we study seed development and how do others study it? First of all, we studied transcriptome and methylome analysis in whole seeds, as you'll see. But then we also did it in different seed parts because the seed is composed of a variety of different tissues that have, as you'll see, very specialized functions. And so we were able to study this in different seed parts by using laser capture microdissection. And you can see that you can do cross sections and you can put them under the microscope. And with lasers, you can cut out different parts. This is a seed right after fertilization. And you can see there's a couple different regions here that I'm not going to go into. And the laser can cut them out, and there they are. You can isolate them. You can isolate the RNA and do transcriptome analysis. And in fact, you can isolate the DNA and do whole genome methylome analysis, as you will see. And so many of you are really not familiar with seeds. And so what I thought I'd do is to show you a little bit of how seeds actually form in a Hollywood-style manner, if you can just bear with me. All starts in the flower. The pollination process sperm going down in the female part of the plant. Fertilization. Formation of a zygote where seed development begins. Beginning of the formation of the embryo. The top part of that embryo will become the plant. The little horns is where all the food reserves are made and stored. Cotyledons. Mature seed. Now the seed is in the fruit. The fruit senesces. Seed falls out. Falls in the ground. And there it is. Thank you. That's the development of a seed. So let's talk a little bit about some of the processes involved in gene expression during the development of the seed. Now this is the summary of about 200 gene chip and RNA-seq experiments, and I don't have time to go into any of the details, so I'm just going to conceptualize. And the idea is, what are the parts of the seed that give rise to specific biological processes that are involved in seed development? How are the genes changing in development with respect to their regulation? And more importantly, what are the transcription factors of the regulators that are in different parts of the seed and that are responsible for the differentiation of those parts and or functioning? So the first thing that we learned was that no matter what the part of the seed is, all of the genes, primarily, not all of them, but most of them are shared in all of the different parts of the seed, no matter what stage of development there is. So different parts of the seed share large numbers of gene sets. That's the first thing we learned. Although there are some specific genes, obviously, because genes have to be able to program unique functions and unique regulatory pathways in different parts of the seed. So you can see that there's some unique aspects to the different tissues of the seed with respect to gene activity. But for the most part, the genes are shared among the different tissues, regions, and compartments of a seed. But there are unique genes that are uniquely expressed in each different part of the seed. And I'll show you some examples of that. The majority, from a developmental stage point of view, also are shared. That is, the genes that are activated after fertilization are utilized all throughout seed development, and in fact all the way up until dormancy, although again each stage has a small number of stage-specific genes. But for the most part, gene activity is activated at fertilization, and what you'll see is that most of the differences are large quantitative changes in gene activity, and we think that those large quantitative changes, along with the very small number of unique changes, is what's really programming all of seed development with respect to tissue, region, compartment, specialization. Now, when you take all of these data, again, chip data, seq data, you can say, well, what is the total number of genes that are required to program all of seed development? And our estimate's about 50,000, 52,000 genes, which in fact is the majority of genes in the soybean genome. We can detect about 54 to 55,000 genes that are active throughout the entire life cycle of the soybean, and most of those are active during seed development. And clearly then the challenge is to try and understand what are the important genes with respect to regulation and what are the genes which are really programming this process. And so not only then can we look at genes at a given stage and see how they're expressed with respect to the different tissues, the different cell types, but you can obviously look throughout all of seed development to try and understand in the different phases of seed development what are the regulatory programs that are initiated and utilized during seed development. And so I'm just going to cartoon this, and then I'll show you some data afterwards. First thing is really important is that during the early stages, when seeds are differentiating, there's a high level of gene activity in very different specific parts of the seed. And these are probably genes that are programming the differentiation of those parts of the seed. But then as the seed switches into a program which is producing food reserves, you can see that there's a lot of genes that are activated during this time period uniquely. So early on, there's a very spatial regulation in a temporal way of gene activity. And then later in development, large numbers of genes come on. Many of them are contributing to producing these storage products. But as you'll see, most of these genes are active throughout the entire seed. And then finally, there's a whole set of genes that are activated at the earliest times of development and are programmed all the way through development, but they have very specific locations within the seed. These are very tissue-specific, region-specific, at least from a quantitative point of view. And I'm not going to talk about this one. So just to show you some data early on, here's some transcriptome clustering data. What you can see is early on you can have sets of RNAs that are active specifically in different regions of the seed. This is the seed coat, and this is a specific region of the embryo. Then as the seed switches into a production period of food reserves, you can see that all of these different parts of the seed, genes are activated coordinately at that particular time. And as I said, many of these contribute to particular products that are being made that are necessary for seeds from a human consumption, but for seeds after seed dormancy and into germination. And then, of course, there's these genes that are activated very early after fertilization, and then they remain active, but they're active in very specific tissues, very specific regions, very specific parts of the seed. And we think that they're not only involved in perhaps the differentiation, but maintaining those differentiated states all throughout seed development. So what was really surprising then is if you look at all of these data, the bottom line is that there's a large amount of functional specialization within a seed. And you can list all the different kinds of metabolic pathways. And so all of the regulatory programs that I showed you apply not only to the general mRNAs, but they apply to enzyme mRNAs, and as you'll see, also transcription factor mRNAs. So these programs are operating on a variety of different genes throughout seed development. And here's the specialization in different parts of the seed. And this even applies to the things that are very important for human and animal nutrition. And so we know where in the seed, in the different cell types, in the different tissue layers, these particular genes and these proteins are particularly made. And then from a regulatory point of view, you can then divide up the seed, and here's right after fertilization, and here's a little bit later in development, and you can see all these transcription factors that are either uniquely present in different parts of the seed or are elevated in different parts of the seed. And so you have a patchwork quilt, so to speak, of gene activity occurring in a very mosaic kind of way, but we've been able to identify important genes, critical genes, that may be involved in, of course, making a seed. Now, clearly, then, one would want to know what are the functions of these genes, what other genes are they regulating, how do they fit together in regulatory circuits in order to program seed development from fertilization to dormancy. So one of the ways we've been able to do this in collaboration with Monsanto, because they're the ones that can really transform plant genes into soybeans, we generated about 750 RNAi suppression lines in soybean, knocking out about 85 unique transcription factors that were localized in different parts of the seed. And just to show you one result, here's a transcription factor that is specific for one layer of the seed coat. This is a very unique layer of the seed coat, and this is involved in protecting the seed when it's in dormancy, but it's also very important for germination after dormancy ends. And what you can see in these RNAi suppression lines of this particular transcription factor, you can see that we get almost no germination. And so downregulating this transcription factor causes a complete loss of the plant in its ability to germinate, and we think then that this transcription factor is very important for processes, either differentiation processes or, in fact, physiological processes that are critical for setting up this part of the seed coat for germination, which from an agronomic point of view or from a plant point of view is critical because without germination, the plant can't survive. And so clearly there's large numbers of gene regulatory programs that are very complex that occur during seed development. And the key, of course, is to try and understand how they all fit together into circuits. Now, from a mechanistic point of view, one of the issues really is how are, let's say, epigenetic changes, methylation changes occurring in the seed that may correlate with any of the gene expression changes that I've shown you. And so we undertook a relatively ambitious project, which has been a little bit technically difficult, but I think we've been making a lot of progress to characterize the whole genome methylomes of seeds from fertilization to dormancy and in every single solitary cell region and compartment of the seed using whole genome bisulfite DNA sequencing, in which you can distinguish between methylated cytosines and unmethylated cytosines at particular parts of the genome. And so the question is, are there methylation changes that are developmentally specific during seed development, and do these correlate at all with any biological processes in the seed, and in fact, do they correlate with gene activity? So I'm just going to summarize about 200 gigabases of whole genome methylome data in a cartoon form, but there's been some interesting concepts that have come out. The first really remarkable thing is that there's an increase, this was very surprising, in the bulk methylation level of the seed from fertilization to dormancy. And in fact, as you go into the terminal stages of seed development and in the dormant seed, the level of methylation of cytosines goes up significantly. And furthermore, the kind of methylation that goes up is in a context of CHH. CHH is the cytosines that are actually the smallest number of methylated cytosines in the genome. But clearly, with respect to going into dormancy, this is the class of methylated cytosines that increases significantly. So that's the first thing that we learned. The second thing, and here is the entire methylome landscape of the soybean genome, every chromosome, all throughout the chromosomes, plotted together in a cartoon at all different stages of development. And so what you can see from this is a couple of things. One, not surprisingly, is that there's a high level of methylation, whether it's CG, CHG, or CHH methylation in regions which have transposable elements. So here's the density of transposons on a given chromosome, and you can see here's the density of the methylated cytosines in those particular regions. And the green are the gene-dense regions, and you can see there's a lot lower level of methylation as you'd predict in these gene-rich regions. And so one can then look at the landscape, the complete landscape of methylation over all the chromosomes, over the genes, transposable elements, and the other. Next thing that you can see is clearly, as I've said, in terms of the CHH context, you can see that there is an increase, a very significant increase across all of the chromosomes. This is whole genome-wide of CHH level methylation as the seed goes from, again, after fertilization into dormancy. And I have some ideas on perhaps what's that doing in a few minutes. And so clearly then, we're looking at changes in methylation in a general sense. And so one can then ask the question, are there changes in methylation in specific regions and specific parts of the genome as development goes on? And so the answer to that is yes. So you can see in just these clusters of changes in methylation as the seed goes from fertilization into dormancy, you can see high level of methylation to low level of methylation, and you can see low level of methylation to high level of methylation, and these are particularly in genes, in specific genes, so there are in fact different changes in different regions. And you can see those on the genome browser. Here's in fact high level of methylation to low. Here's a low to high. And in fact, if you try and correlate this with RNA activity, there's really no correlation. You can change methylation patterns in specific genes and correlate those with changes in RNA accumulation, or you can change those and have no correlation in terms of RNA accumulation. And in fact, if you look at the landscape around the genes that are the most highly expressed at a particular time in the development of the seed, and these are these storage protein genes, what you can see is in fact that there's almost no methylation around them whatsoever. In fact, there's no changes. And so these genes are being activated transcriptionally and then being repressed transcriptionally in the absence of any methylation changes during seed development. Okay, let me just wrap up two concepts and then I'll close. One of the most important things about seed development is that you get a change in DNA content. You get an endoreduplication. And the endoreduplication starts from one side of the seed in the beginning and then goes towards the other side of the seed. And we know that this is correlated with a large increase in the activity of seed protein genes, that is, the increase in DNA content. Endoreduplication essentially produces more copies of genes that are then necessary for huge amounts of gene activity, huge amounts of mRNA, huge amounts of storage proteins being produced, because 40% of the dry weight of the seed is in fact storage proteins. So the question then becomes, and in fact, in the part of the seed called the axis, which becomes the plant after germination, the question really becomes, is endoreduplication and methylation coupled or uncoupled? And so we use both laser capture and microdissection to get out different parts of the seed. This part where you have large increase in DNA content, this part where you have very little increase in DNA content at that particular stage. So we compared the whole genome methylomes of those particular parts of the seed, and we also compared the axis, which does not undergo endoreduplication, to the other part of the seed called the cotyledon, and the answer is that you don't find any correlation, meaning methylation is maintained during this endoreduplication period, which is one of the most important periods in seed development. The last thing really is, can you see in a crude way differences in different parts of the seed? This part of the seed will become the seedling, the plant after germination, and this part of the seed will senesce and go away. Is there any changes in the whole genome methylome in different parts of the seed? And what you can see is, again, the answer is significantly yes. We're getting a large change in CHH level methylation. And in fact, in the seed coat, as you can see, compared to the rest of the parts of the seed, the CHH methylation is very low. You can see right here it's much lower than in this part, and this part of the seed. So this says that different parts of the seed have different levels of methylation on a whole genome basis, at least with respect to CHH methylation. And now I'm sticking my neck way out. The question really is what does this mean? What does it have to do from a functional point of view? And so sticking my neck out, you have to think that after dormancy, the seed germinates and produces the entire plant. And that means that anything that happens during germination to inhibit that process is going to be lethal to the seed. And so one of the things that might be happening is that the low level of the decrease in CHH methylation in the seed coat might be activating the small RNA pathway. And small RNAs have been shown to reinforce methylation in transposons to downregulate their activity. And there's some very nice data in plants that show that small RNAs can move from one part of a plant to another. And so I'm totally speculating here that maybe the reason for the very low level of CHH methylation in the seed coat relative to the embryo, which becomes the whole plant, may be because small RNAs may be moving from this part of the seed to this part of the seed, and then completely tampering down transposon activity so that when the seedling develops, we're not going to have any interruption of genes in any way and allow seedling development to occur perfectly normally. And so just finishing then, what our data have shown is that the seed is highly specialized with respect to gene expression programs in different parts of the seed, particularly with respect to transcription factors. And one can get at the properties and functions of those factors by using suppression experiments and others. And then what I hope I've showed you is that there are large changes in the methylation landscape across the entire genome throughout seed development. Most of those changes do not correlate with changes in gene activity, but maybe some of those changes have something to do with perhaps protecting the subsequent plant following germination. And so just to finish then, these are the people in my laboratory. That's on the deck of my house in Topanga in Los Angeles, and that's in our lab. And this has been a collaborative effort between my laboratory, John Harada's laboratory at UC Davis, Matteo Pellegrini's laboratory at UCLA, with help from Monsanto, a lot of help from Bob Fisher at UC Berkeley, and support from the National Science Foundation. Thank you very, very much. I appreciate you.
Thank you, Bob.
Thank you.
Questions?
So the methylation study is really amazing. And it's particularly, you show that there's a difference between CpG and CHH and CHG. And I assume that these the different methyl transfer responsible for the maintenance or change of methylation on these different kind of contexts. So I was wondering whether, since you have RNA-seq data, have you looked at the activity of these genes and the spatial distribution of these genes along the seed?
That's a really good question in terms of looking at the genes that are involved in methylation. And the answer to that is that I think that the process is complicated by just the way seeds develop. And a lot of RNAs hang around for a long time during the development of seeds. And so when we look at these particular messenger RNAs, we really don't see large differences from a temporal point of view or from a spatial point of view. And we're really not sure exactly what that means in terms of contributing to the methylation landscape. And we have to explore that further because it's possible that there could be translational controls on top of this that we're not seeing. But there's no obvious thing that you can see by simply looking at the RNA accumulation.
Bob, I'm curious what you can say about, so you talked about the soybean seed as a model. What can you say about how this carries over to the monocot seeds, the grass seeds?
You know, I don't really know very much about monocots, but what I can say is about dicots, and that is Arabidopsis, because we've done a very similar series of experiments comparing gene activity in every part of Arabidopsis from fertilization to dormancy, and we see exactly the same principles. Mm-hmm. And you can identify, obviously, some of the same genes, but that's not surprising because they're kissing cousins and only about 150 million years apart. But I would suspect that you're going to find the same thing for a rice or a corn or whatever, because these plants aren't that far diverged from each other. But I don't think anyone's done that particular experiment. Thanks. Question over here.
So based on your gene expression data in the different compartments and your knowledge of the kinds of oils and lipids that are produced in the different compartments, are you able to reconstruct any of the metabolic pathways that are used to produce the diverse set of metabolites?
Yeah, we can do that.
Okay.
It's a lot of bioinformatics, but we can definitely do that. Most of these pathways, of course, exist in most parts of the seed, but they're elevated in very particular parts. And you can reconstruct those things quite readily if you're willing to sit at the computer for any length of time. And then, of course, what's regulating them, you can try and correlate that with the transcription factors that are those parts of the seeds and make those trines. It's just there's a ton of data here. And hopefully all of you will be using those data, because in a couple years at the most, I'll be sitting on the beach in Malibu and reading books. So the point here is there's a lot of data for people to be able to really understand the development of the soybean seed. There's a question over here. Yeah, so the question is, what's happening with respect to endoreduplication of the genome after germination? So the endoreduplication is specific for only the part of the seed that's involved in storage product production, the cotyledons. And after germination, those cotyledons are destined as an S and die. And so the part of the plant that becomes the part of the seed that becomes the plant, which is the thing called the axis, which is a very small part of the seed, there's no endoreduplication in that part of the seed. And so therefore, the rest of the plant, as the seed germinates, that the roots and the stems, the new roots in the stem, come from this axis, and endoreduplication is not playing a major role. The endoreduplication is in the cotyledon because the cotyledon only has a small amount of time to produce a tremendous amount of oils and proteins with very few genes to do it. And so it's increasing the copy number of those genes by increasing the entire genome over a very specific period of time. And so after the cotyledon's dead, that endoreduplication is obviously no longer a factor.
Thanks, Bob.
Thank you very much.